I'd like to thank the organisers for the invitation to speak to you today about our point-of-care diagnostic test, and it's great to hear all the amazing progress being made on hepatitis elimination globally. So at the Burnet Institute, we proudly acknowledge the Boonwurrung people of the Kulin Nations as the traditional custodians of the land on which our office is located. We pay our respect to elders past and present and extend that respect to all First Nations people here. So we know the global challenge of viral hepatitis is huge, and I don't need to remind you about these, but we've been making amazing progress, but there are still challenges to meeting those targets that have been set by the World Health Organization and in the elimination of viral hepatitis. Now, the World Health Organization recommendations for hepatitis B mention that ALT levels should be considered in some of the diagnostic decision-making that's done. And this has led us to pursue whether our point-of-care diagnostic test for measuring ALT would be useful in these situations. Now, why would you need to measure ALT in a point-of-care diagnostic when we have fantastic laboratory facilities in most countries for measuring ALT levels? Well, some of the problems are that this requires a venous blood draw, it requires pathology lab infrastructure, and there are delays in receiving results from hours to days in some cases, and this can really impact timely decision-making. This slow turnaround increases the risk of patient loss to follow-up. Now, there are multiple reasons why you would want to measure ALT. Apart from viral hepatitis, there's also elevations of ALT through alcohol consumption. There's also a use case that we're exploring around drug-induced liver injury. Paracetamol is one of the most common reasons people call the drug the poisons information line in Australia and in other countries. But there are also many drugs on market that require regular monitoring of ALT levels with patients on those therapies. And there's also fatty liver disease, which can lead to elevations in ALT. But there are certain scenarios where in low-income settings, particularly in remote and rural locations, access to pathology services are limited, and a point-of-care test could be very useful for timely diagnosis. In resource-rich settings, the market is around monitoring of drug-induced liver injury and potentially ALT levels in resource-limited scenarios. So ALT is an enzyme that the liver produces, and its primary function is to transaminase alanine, remove an amine group to create pyruvate and glutamic acid, and this is key to our metabolism. But when the liver gets damaged, ALT is released into the bloodstream, and it's used in this way to detect damage to the liver. Now, what people sometimes don't appreciate is that there's actually multiple forms of ALT that are produced in humans. There's ALT1, which is the predominant form produced by the liver, and more than 90% of the ALT that's circulating in the blood is ALT1. But there's another form that's ALT2, which is actually produced by the liver as well, but it's also produced more by muscle and heart tissue, and so it can become elevated through strenuous exercise and also in heart attacks. And the enzymatic tests, of course, measure ALT1 and ALT2, so they're not specifically measuring the form of ALT produced by the liver. So to develop our point-of-care test, which is based on a lateral flow setup, we developed a target product profile where we had some conditions we wanted to meet, that the test would be a simple-to-use test that could be performed easily by a healthcare practitioner, that could also be used by as a self-test for lay people. Ideally, it would be a quantitative test, so you'd actually get international units per litre from the test, but it could also be semi-quantitative through the use of a card. We also wanted to have a very small volume of finger prick blood. A lot of tests require 50 or 100 microlitres of finger prick blood. If you've tried to get that much volume, it's actually quite difficult. So we wanted to have a maximum volume of 20 microlitres and a preferred amount of 10 microlitres. And the test should only take a maximum of 20 minutes, preferably 15 minutes to perform. The other requirements are pretty standard for diagnostic tests: stability for at least a year, preferably 18 months or longer, and use in different temperatures. So what we had to do to develop this test was actually develop very specific and sensitive reagents to capture the ALT in blood. And what we did was produce rabbit monoclonal antibodies that we coupled to either a fluorescent or a calorimetric substrate, and these are placed on the sample pad. On the test line, we made nanobodies in alpacas to ALT1 and engineered them so they would be highly specific to the form of ALT1 that's produced by the liver. And when the sample of blood is applied to the sample pad, these react with the rabbit antibody that's coupled to a fluorescent or a calorimetric bead. This running buffer then flows across the sample pad and it's captured on the test line, and the excess material becomes the control line. And this is different to the previous version that we tested in the lab, which Jess Howe published here in this paper, if you'd like to read it. The reagents are different, the schematic of the test is different, there's no reference line. And the reason we did that redesign was really to make sure that we could manufacture hundreds of thousands, if not millions, of these tests in the future in a quality management system under GMP conditions. So we've kind of met all of those requirements now and have a test that we can actually go forward with. The test itself was designed to fit into this cartridge here, which is here if anyone wants to see it afterwards. I did a test on myself this morning after a few glasses of wine last night, and can happily report my liver's fine. What it has in this cartridge is an integrated lancet. So the lancet is retracted when you take it out of the packaging. You press it against your finger and it pricks your finger. It's extremely painless and you start to bleed. This little blood collection unit here takes 10 microlitres of blood. It's precisely measured. You then rotate that onto the sample window. It delivers the blood into the sample window, and you press a button on the cassette, and the blister inside the cassette contains the running buffer, and that initiates the flow. So there's no separate lancet, there's no separate dropper bottle, there's no chance of needle stick injury. It's very fail-safe and easy to use for even lay people can use this test. And the result is interpreted in 20 minutes with this tiny little handheld reader. It fits in the palm of my hand and it's battery operated. And this provides a quantitative result. So this is the analytical performance of the test, the fluorescent version's here and the coloured version's here. You can see it is linear between about 0 and 4 to 500 international units per litre in the way we've tuned it at the moment, and the fluorescent and the visible version are very similar in their performance. And we can tune this test to measure different ranges depending on the concentrations of reagents that we use. We've done a study with stored blood that we actually reconstituted back into whole blood. It's very hard to do finger prick studies in the lab, as you can appreciate. So what we do is we take stored samples and add red blood cells to make them venous blood effectively. And this is the correlation between the antigenic and enzymatic ALT. And we get a very good correlation, but you will see there are some samples that do not correlate, and I'll get to those in a minute. So again, the fluorescent and the visible tests are very similar, very high correlation with enzymatic. And if we do a receiver-operator characteristic curve, you can see the area under the curve is about 0.88 for both tests. We then also looked at the sensitivity and specificity at 40 international units per litre using these stored samples, and we have 75% sensitivity, 88% specificity, 88% positive predictive value and 75% negative predictive value. So it's performing very well. We still have some questions. These are some of the participants that we looked at where they had longitudinal samples collected. And you can see that the trend over time is very similar for each of the people, with this participant having a perfect one-to-one relationship between enzymatic and antigenic ALT, whereas some participants have, although the trend is the same, the relationship appears to be different. So, for example, in participant 3, the antigenic ALT level is higher than the enzymatic level. We don't know why that is. Speculate that sometimes the liver can produce a lot of will release a lot of ALT, but not all of it is enzymatically active, so potentially we're underestimating the amount of ALT that's being released by the liver. In other participants, we've got a higher amount of enzymatic ALT than antigenic ALT, and this may be because we're only measuring ALT1, we're not accounting for ALT2, whereas the enzymatic assay measures the total amount. To look at that more specifically, we're actually about to get monoclonal antibodies to ALT2 that we can add into the lateral flow design and capture the total amount of ALT that's produced by the liver. So this is the 4 versions of the device that we have at the moment. We've got the fluorescent version, a visible version that both use the reader. You can also do this in a reader-free test as a semi-quantitative test using a scorecard to interpret the result. And we can also do this in a standard cassette if that's preferred, which is sort of the ultra low cost option, but you will have the complication of needing a separate lancet and dropper bottle and the error that could potentially creep into doing the test that way. Currently we're developing the test. We're doing a couple of clinical studies. We've got a drug-induced liver injury trial that's part of a phase 3 sub-study with a pharmaceutical company in the US. They've got 6 sites that are using this test, looking at its use as a fluorescent version of the test. And we're also doing a multi-site clinical study with Jess Howell and her team at St Vincent's Hospital and Monash Health, looking at the performance of the visible and the fluorescent version in patients attending the gastro clinic there. And of course, in discussions with Jibril about the use of this in the studies planned in the 3 African countries. So in short, we've successfully developed this test. We're poised to do more clinical studies to evaluate its performance on finger prick blood and really look at the sensitivity, specificity and the correlation between enzymatic ALT further. Because this is a new biomarker, we need to provide this data for future regulatory submission as well to justify that we're measuring a valid thing. So all of this work was done at the Burnet Diagnostics Initiative. I thought I'd just briefly introduce this to you. We're a facility at the Burnet Institute that has an ISO 9001 certified quality management system. We develop diagnostic assays such as lateral flow, but also multiplex bead assays for large-scale serosurveillance studies, which we're currently doing, ELISAs for different pathogens and biomarkers, and we do analytical and clinical studies to verify and validate different diagnostic tests. And I thought I'd just quickly sneak in a little slide about syphilis, because I know it's really central to triple elimination. This is another test we've successfully developed. There's a lot of parallels with syphilis and hepatitis C in that the more people who are cured of hepatitis C, the less useful the rapid diagnostic tests are for antibody measurement, because you'll get a lot of false positives with people coming up positive because they've had hepatitis C in the past. The same is true for syphilis. If you've ever had syphilis and had successful treatment, you will remain antibody positive for life. And so the rapid diagnostic test for syphilis can't distinguish a current infection from past treated infection. And so to address this, we developed a new rapid point of care diagnostic test in the same cassette that is able to detect primary, secondary and early latent syphilis with reactive RPRs at 100% sensitivity. And the big difference here is the past treated, 31 out of 41 past treated cases returned a negative result. And if we exclude people who had syphilis in the last 3 months, the specificity increased to 83%. And in the never infected population, the specificity is 99%. Now, if you compare this to the 2 currently available diagnostic tests for syphilis in Australia, which are both made by Abbott, you can see that our sensitivity is very similar, but the specificity is completely different. So in those tests, in the determined test, every single past treated person came up as positive, whereas ours did not. And so you would be 5 times over-treating people if you were relying on the currently available tests for syphilis. So we think this is a really significant advance to really help address the problem of syphilis and in triple elimination. This test has been licensed to Atomo Diagnostics and we're currently doing a technology transfer for manufacturing and getting ready to do a clinical validation study for regulatory approval. So I hope that's exciting. These are all our collaborators. This is a really large piece of work. There's been many, many people, but a really big call out to Lillian Hall and Helen Wire, who've really led the development of this test in the lab. Our collaborators at Lumos and Atomo, of course, Jess Howe, who's been a wonderful collaborator and supporter of this test, and our other clinical collaborators. Without them, you can't develop diagnostic tests. You really rely on support from clinical groups to find the patients and recruit them into your study. And I'd also like to really thank the participants of this study who volunteered their blood and time to help us develop these tests. Thank you.